Hey everyone! Hope you had a nice summer. So what I'm going to be talking about today is topic 9.1, which is transport in the xylem. So to start off, this is a cross-section of a plant stem, and so as you can see, in the vascular bundle, it consists of the phloem, the cambium, and then the xylem, and so I'm going to be talking about the xylem. And the xylem transports water and solute from the roots to the leaves. This movement of water is passive, and heat from the energy is necessary for this process to occur, as it provides the energy required for the evaporation of water. Now, this is a cross-section of a leaf, and there are two mesophyll layers. There's the palisade mesophyll, and then there's the spongy mesophyll, and that's where the veins or the vascular bundles are found. So transpiration, first off, is the loss of water vapor from the leaves and stems of plants. And so when a leaf's guard cells open, they're right here, to exchange gases, some water in the form of water vapor is lost. So the stomata, or pores, so this would be the stoma, are simulated by hormones. And so, for example, abscisic is the hormone used to close the two guard cells. And so this transfer of gas, carbon dioxide, and oxygen is necessary for photosynthesis to occur. Once water has been taken up by the roots of the plant, it's pulled upwards into the leaves where it then evaporates. This flow of water from the roots to the leaves is what's called the transpiration stream. So loss from the leaves draws water vapor from the spongy mesophyll layer into the airspace. Water in the mesophyll, moving water from the mesophyll layer to the airspace, draws water from the end of the xylem into the mesophyll layer, and then with cohesion, this movement of water molecules extends all the way down the xylem, and that creates the suction effect. So mature xylem vessels are long, dead structures which are made up of cells arranged end to end with no end cell wall. Notice how there isn't like a separation of cells here. The cytoplasmic content also dies to perform a continuous tube. The cells also lack a plasma membrane, which allows water to enter the vessels freely. In addition, they also contain pores in the outer walls, which allows the movement of water out of the vessels and into the surrounding cell leaves. The outer cell walls contain thickenings, which resemble spirals or rings impregnated with lignin, which makes the vessels strong and able to withstand low pressures. And low pressures would be, for example, the suction. So the low pressure causes more water from the roots to be pulled upwards through the xylem tubes. This is called the transpiration pull. Transpiration pull works due to the cohesion of water molecules. Hydrogen bonds form between the water molecules, allowing the formation of columns of water which are not easily broken by low pressure. Adhesion also plays a role in maintaining the transpiration pull. The water molecules adhere to the walls of the xylem vessels, preventing the columns of water from breaking. So the cell walls of the xylem vessels are charged, which attract the water molecules, and the adhesive attraction of water to xylem vessel walls moves them up the stem against gravity. Adhesion also helps prevent the columns of water-filled xylem vessels from breaking. And the concentration of mineral ions inside the plant's roots is actually 100 times higher than that found in the soil. So mineral ions, for example, Cl negative, have to be transported into the roots via active transport. Protein pumps exist in the plasma membranes of root cells, like this one seen here. There are many types of these protein pumps for the absorption of many different mineral ions. Active transport requires ATP production, which we learned about, and therefore the roots cells also contain many mitochondria. The branching of roots and the formation of root hairs increases the surface area for the absorption of mineral ions by active transport. Water will move into the plant roots passively to try and achieve a concentration equilibrium of the mineral ions in the plant and the mineral ions in the soil. So pressure in the xylem will then increase, and that will push the water up through the rest of the plant. So now I'm going to talk about some water stress habitats. Some examples are deserts, which have low precipitation and high temperatures. Also places which have high altitudes and also high latitudes because they have low precipitation rates. Also the tundra, water there is in the form of ice or snow. Also areas with sandy soils because water drains rapidly. And also shorelines or areas with high salt levels. So here are some adaptations to make these areas habitable. One of them is waxy leaves, and this is the development of a waxy cuticle on the upper and lower epidermis, which prevents water loss. Also being succulent, leaves are sometimes reduced to needles, and the stem is quite fleshy, where the water is stored, and is also the main site of photosynthesis. Also, there's reduced number of stomata. I don't have a photo for that. There's also reduced leaves, which in the hopes of gaining a smaller surface area. Firs and pines do this, where they develop needles to reduce the surface area, and these needles also have a thick, waxy cuticle. Finally, grass in sand dunes have a thick, waxy epidermis. They also have a specific adaptation which helps them, where their leaves are rolled in this formation. This way the stomata are in an enclosed space and are not exposed to the wind. A rolled leaf also acts as a channel for water to drain to the specific root of that grass plant. So one of the things that we're going to learn about is modeling the transport in the xylem using simple apparatus. So we're going to watch a bit of this video.
The level of water in the capillary tubes is higher than the level of water in the beaker. The narrower the capillary tube, higher the water level.
So hopefully that explains something in case it wasn't quite clear. So now I'm going to talk about plant factors that affect the rate of transpiration. So first off is the leaf's surface area, also the thickness of the epidermis and cuticle, stomatal frequency, stomatal size, and stomatal position. These will all affect the rate of transpiration in plants. So speaking of the rate of transpiration, we can actually measure how much water is taken up by the plant with a potometer. And a potometer measures the volume of water taken up in usually centimeters per second. So either the measure is given a number of centimeters at a time or set a certain time and measure in centimeters. So what this basically means is, so this is the capillary tube, it's filled with water. And so if you make a bubble in it, for example right there, then you can actually measure with time how far that bubble travels and therefore how much water the plant has taken up. So if you start it, for example, at the 10, and you say, okay, I'm going to wait until it gets to the 7, and then you measure how long that takes in time. Or you could say, okay, I'm going to let it sit for 15 minutes and see where it goes from the 10, and then you'll measure it in centimeters, the change. So that's just how that works. So the rate of water loss from the shoot can also be measured under different environmental conditions. Now, I'm going to talk about those, but first there are some limitations to using a potometer. One of them is cutting a plant shoot and then inserting it into the tube could damage the plant. And also the plant doesn't have any roots, so there isn't any resistance to the water being pulled up, which is something to think about. And so some abiotic factors that affect the transpiration rate in a typical terrestrial plant is light intensity, where the rate of transpiration is much greater when light is available because the stomata actually close in the dark Also the relative humidity, where so air inside the leaf is saturated, and the lower the relative humidity outside the leaf, the faster the transpiration rate because of the steeper concentration gradient. Also temperature. There we are. Okay. As temperature rises, so does the rate of transpiration. This is because higher temperatures provide latent heat of vaporization. It also increases the kinetic energy, so there's faster diffusion. And also it warms the air, so the concentration is lower, which increases the concentration gradient. Also air movement. The wind increases the transpiration rate because it removes the humidity around the leaf produced by transpiration, making the concentration gradient steeper. Atmospheric pressure is another one. Decreased atmospheric pressure increases transpiration. And finally, water supply. There's little transpiration if there is little water supply in the transpiration stream. This is because transpiration relies on the mesophyll cell walls being wet. When cells are flaccid, the stomata close. So, yeah, thanks for listening, you guys. Bye.